This is a production of Cornell University.
Thank you, Don. So obviously we're going to talk a little bit about berries today. That's what I do, and that's what I've been doing at Cornell now for 17 years. So I work in actually several different species, as you can see up here: raspberries, strawberries, and blackberries. So Cornell has a long history of work in berries. At the station opened in 1882, and berries were one of the first fields that were planted that first year at the station in Geneva. Of course, at that time, the station was an independent unit, was not part of Cornell, but did join Cornell in 1923, and that program has been maintained through that time. We are the oldest berry breeding program in the United States, and as far as continuously active, as far as I can tell, we're the oldest continuously active program in the world, but those stats are a little hard to come by, so there may be some dispute there. Over that time, our program has developed 41 different raspberry varieties, 3 of which I have named and patented. You can see them there. We've also developed 43 strawberry varieties over that time, the 43rd one coming out today, being announced today in the Chronicle. We did plan that a little bit. So, but so generally, and then there has been some blackberry varieties. There's been little work in blackberries, mainly because New York is generally too cold to consistently grow blackberries, but that's changing with some new genetics that we're using in blackberries. So my program and my appointment is research and extension. We have a very diverse set of clientele that we work for, that we try to help. Certainly the home gardeners. Gardening is the #1 hobby in the United States. Of course, can be done by people of all ages, and people continue to do it throughout their lives. And so our varieties are certainly, many of them are suitable for home gardeners, and they really love berries. There's nothing better than a berry out of your backyard. It will be the best tasting berry you'll ever get. That said, you can't grow berries in your backyard all the time. So we have the people that cater to those home gardeners, certainly the retail nurseries. These are companies like Burpee Seed Company, sell our varieties. The Gardens Alive Company, which is Gurney's and Michigan Bulb Company, and some of those other catalog companies that sell directly to the retail market. Generally small numbers, high price per plant. You get into the commercial nurseries. You might recognize nurseries like Norse Farms, Crone Farms in Michigan. There's a lot of berry nurseries around the country. We work directly with them with our varieties, licensing our varieties, but also working with them with new material. They propagate some of our material to give out to growers for grower trials before they're released. And so we have a very good relationship with many of these growers around the country. And then the commercial producers, both domestic and international. We work directly with the New York State Berry Growers Association. They provide member growers who will test some of our material before it makes it to the marketplace. As well as larger companies in California that want to see things pre-commercialization so they can be ahead of the game when it comes out. We have international cooperation as well. So we have exclusive contracts in Mexico, in Europe, and we're working on a few other countries so that they can see our material very quickly, and if there's something they like, they can bring it to the market before everybody else, and they can have it for their own programs, for their own production. So in the extension side of things, we do things like variety trials generally, and a lot of production technology research. I worked quite a bit with Marvin, of course, on that, Marvin Pritz, on that area. This was a project that Marvin led on low tunnels for strawberries with Day Neutral to extend the season. It's been very, very successful, and the berries look really good under the system. And we think it's a very good option for the New York grower. So right now there are strawberries available underneath these low tunnels. People are still picking strawberries where they have them, and they can pick strawberries generally through the mid-November is how we find it. Often we just run out of berries as it cools down, but the quality and the season extension is really great using some of these things. Of course, we started using high tunnels at the station in 2008. We put up our first set of about a half an acre of these are Haygrove high tunnels. You can see they're multi-bay, they're interconnected. These are seasonal, so the plastic comes off in the wintertime, goes back up in the spring. And for raspberries, this step in production technology has really changed the raspberry industry. All it does is keep the rain off. But a raspberry that gets rained on in the field has a shelf life of about 2 days. It takes 4 days by truck to get from California to New York. So if they get rained on, even though it doesn't rain a lot in California, it does rain sometimes, although in the last few years, not very often, those fruit are unsellable. Put them under tunnels where they don't get wet, you get a shelf life of 10 to 12 days. So there aren't very many technology you can use to get a 500 to 600% increase in your shelf life. So it's really important for raspberries because they're so perishable. We also find the plants grow better, the fruit's bigger, and the yields are higher. All good things.
But you have to irrigate.
You do have to irrigate. For the commercial market, for the supermarket in California, all raspberries are grown under high tunnels. There's no open field production. That way they can guarantee the quality and they can provide a consistent product that we expect to see in the supermarkets every day now. Doesn't always work out the way you like. This is Hurricane Ike in 2008, about a day and a half after we finished putting the plastic on. And it came right over the top of us, 70-mile-an-hour winds, and tore it up pretty good. 2 weeks later, we had it rebuilt, put back together. So it looked really bad that day. We disassembled it, the parts that were bent, straightened some of them out, replaced some of them, and we were back in business in a few days. So it really did look bad Monday morning, though, coming to work. It was pretty disheartening. We hold field days then, some of the variety trials. We have all the fruit. Might as well bring people in to taste it. So you can see we put all this fruit out so that people can sample it, see the difference between the varieties. Growers come in, they see how we're growing it. It's just a way to get direct outreach and to get direct input from the growers on what they like. So some of you who came early, you saw in the back, I put out some raspberries. You saw there was a little score sheet too. I figured I have a captive audience. I might as well get some information out of it. But that's the type of thing we'll do. We'll provide grower samples, let them taste them, let them rate them on what they think about raspberries, or strawberries too. And it turns out people don't all like the same thing. Generally, the good tasting varieties do separate out that most people like. And then there's often the outliers, people who like tart berries, the really sour ones, and that's fairly common in raspberries to have acidic fruit that is tart in flavor. But generally the market has moved towards sweet. So higher sugar, bigger and redder. Hopefully Colin was able to bring some fruit down in Geneva and they got some to eat. I didn't put any score sheets out for them, but I didn't want to leave you out for fruit. If you're not seeing it, it should be there, and if Colin didn't bring it, then I'll talk to him later. Colin is my technician, so it's how I get anything done. A little bit about the statistics of berries since I've been at Cornell. So when I came to Cornell, the strawberry industry in the United States as a whole was about a $1 billion industry on about 46,000 acres. The vast majority of those acreages are in California. That part has not changed. Now there's about 59,000 acres. This was 2015 data. 40,000 of those acres are in California. The other 19,000 acres are spread across the country, about half of that in Florida, and the rest spread out around the country. New York is fifth in production, in acreage in strawberry production in the U.S., and that is a very small fifth. $2.2 billion in the U.S. in farm gate value. 1.8 billion of that comes out of California. It gives you an idea of the scope of the industry there. It is a year-round industry. They are producing strawberries pretty much somewhere in California, pretty much year-round now. Raspberries is really where we've seen the real dramatic changes since I've been here. So in 1999, the process industry has stayed the same. That's pretty much Washington, a little bit in Oregon. The values has gone up a little bit. You can see 104 million in 2015 on a slightly fewer acres. That is a machine harvested industry for frozen primarily. It's very concentrated on 3 or 4 varieties that they use. Summer, produced in the summer with a very specific training system for the machine harvest. Where we see the real advance is in the fresh market. In 1999, California had about 1,800 acres of raspberries. In 2015, it was up to 8,800 acres. For fresh market raspberries, the USDA only takes statistics on California. That tells you what The rest of the country doesn't even register in their statistics. And the value has risen from $31 million in 1999 to almost $500 million worth out of California. And that's just California. If you think about where raspberries and strawberries are grown, we have Mexico just to the south, and there's a tremendous amount of fruit that comes from there as well. As far as New York, the industry has been flat for pretty much the last 20 years worth of statistics. In '99, we had about 1,600 acres. In 2015, they only registered 960 acres. Across the region, that is the top states that produce the varieties that we produce here, that cold climate, Michigan, Ohio, Pennsylvania, and such, there's about 6,800 acres in '99, now only about 3,800 acres. As far as the statistics that it collects are concerned, the value stayed pretty much the same. So what's changed? Obviously, raspberries, we have seen a 5-fold increase in acreage and a 15-fold increase in value in fresh red raspberries. And it is specifically fresh red raspberries we're talking about. And now they are shipped. You go to any supermarket pretty much any day of the year, you'll see raspberries. Of course, you know the names. Driscoll's is the biggest name in raspberries, and they have driven that industry tremendously. There's increased berry production in Mexico as well. The vast majority of blackberries are coming out of Mexico. There's a particular variety that works very well, can be manipulated to produce fruit most of the year. Of course, labor is very affordable there, and they can ship the berries. They hold very well, and they're shipping them into the U.S. But in the U.S., blackberry production has not increased nearly as much as the other berries, although we're starting to see that change. The thing that's changed for New York is the locally grown movement. In '99, there were only about, or in 2000, there was only about 230 farmers markets, only I say, in New York. Now there's over 500, and that trend continues. Nationwide, that trend continues as well, over 7,800 farmers markets. You go to farmers markets in this area, there's almost always berries, locally grown, but those acreages are rarely caught in the statistics because they're very small acreages, a half acre, a quarter acre. A quarter acre of raspberries could be a lot of raspberries to take care of. And so sometimes they don't even make it into the statistics because when those sheets come out that they have to fill out, and they're growing 14 different crops because they've got green beans and they've got pumpkins and they've got sweet corn and they've got everything else they're growing, the berries don't even register on their statistics. So we know people are growing berries in New York. Our beginning berry growing workshops that we hold are packed with people excited about growing berries. But you can do it on a very small scale and still produce enough berries to have some money, to make some money. And so it's just one crop within a many crops that many of these farmers are growing. So these small grower statistics are really difficult to capture in the USDA's overall statistic picture. The other thing that's changed then with these farmers markets and the locally grown movement is that people are more interested in distinctive varieties for these markets. The supermarket has gone towards one thing, big and red. That's what they want, big red raspberries, big red strawberries. And that's pretty much where the marketplace is when you go to the supermarket. You know, they look great in the container, but sometimes the eating quality is not where you'd really like it. When you go to a farmer's market, you want a high quality berry, you want a high flavor berry, and you don't want what you can get in the supermarket. If I want a big red raspberry that doesn't taste very good, I can go to the supermarket. So when people go to farmer's market, sometimes they want something a little different. So in my program, and of course we still want to produce a big red raspberry because our California collaborators, they still want those for the supermarket. So this is a variety we put out a few years ago. Big and red. Flavors adequate. Ships very well. So you got to balance what you're trying to do with the variety. You know, with strawberries, this is Archer. If you haven't, you'll see some pictures in it in the Cornell. It's a very large red berry. This one happens to be very high flavor as well. It's a little softer than what you'll find in the supermarket, so we're hoping for the local market people will enjoy this as well. Nice to pick. It fills containers really fast. People really like that, both for U-pick and for pre-picked. But we also have some darker berries that we've put out. So here you'll see on my right is Purple Wonder that's being sold through Burpee. It's much darker than an average strawberry would be. And that color carries through all the way through the flesh. So it is a dark fleshed berry as well. The juice comes out of it looking like wine. It's that dark. But the commercial market really doesn't know what to do with something like that. It doesn't fit into any of their marketing schemes. So you see Burpee has a nice display on it at their website. As you'll notice over here, they're selling them for $10.95 a piece per plant. The commercial grower buys strawberry plants for about 13 to 15 cents a piece. So it gives you an idea of what the retail market will bear when it comes to specialty varieties. I had no part in deciding what the cost of these would be. Burpee did that on their own, but we get a very small portion of that as royalties back to the university.
Courtney, I'm sorry, but why can that not be shipped to all of those states?
There are regulations on shipping plants sometimes. California certainly has a lot of regulations on you can't ship certain species. I don't know about all of these states. Some of those are not states, so there must be some territories involved there as well. And it may be they don't want to ship some of these places as well. But certainly California, you have to inspect plants, for instance, for Japanese beetles before you can ship plants there. So there are things like that. But that would be my guess there. We also have a white strawberry. So that's one there you see in the catalogs listed as pineberry. White flesh, red seeds, tastes like a strawberry. Really just a novelty, but it's interesting. Really not something for the commercial market, for the retail market. It fits something that people like to do because they like to tinker in their gardens. They like to pick stuff and show their neighbors. Look what I did. You've never seen anything like this. So it's very popular in that kind of market. But again, it's a very small portion of what we do. Some of the darker raspberries, Crimson Night. That was the one I put over on the table with the refreshments. Dark fruited, very sweet, freezes very well, much darker than what you see in the supermarket. And then a peach colored one, Double Gold. Again, novelty. It's what people like. Flavor's very high. It's a little on the soft side if you had some back there. So it doesn't ship particularly well, but it eats well, and people really like it. And in your backyard, it's very interesting to look at as well as eat. We work a little bit in purple. Primarily, we're working in purple because we're trying to transfer traits from red into black. Sorry. When you cross a black raspberry and a red raspberry, the fruit's purple. So we're trying to transfer thornless, which we have done, and we're trying to transfer fall bearing as well, which is in the middle stages for that. So some of the stuff we're looking at, but if we were to get a purple that was interesting, we'd certainly there is some market for it in the retail market. Yellow, there's lesser interest in yellow, pretty much a novelty, makes it look interesting when you have a fruit salad, but they pretty much taste like red raspberries, so most people will choose red when you give. But we want to put out a yellow one just to have a complete suite of varieties. This is not one we're working on. This is what you'll find on the internet. People say, oh, I want blue raspberry flavor. I say, that's not a real flavor. So these are not the kind of berries we're growing, and we really wish that blue raspberry flavor would go away. But it is something I actually do get asked about fairly often. So the breeding program today is traditional breeding. We crossbreed, we choose parents, transfer pollen, collect seed, grow out plants. It's a very straightforward process that's been going on for centuries. We work in strawberry, both short day, which are the June bearers that we grow mostly in New York, and the day neutral, that more interest that will provide that season extension in those low tunnels. Raspberry, both black and red. Again, the floricane is for summer production. Start somewhere in June, will produce through most of July, and primocane starting in July and produce into the fall through till it gets too cold. With blackberry, I mentioned traditional blackberries fruit in the summer. They don't do well in New York because they generally freeze. The canes freeze, and you don't get a reliable crop. So what John Clark at University of Arkansas started this a number of years ago to work with blackberries that would fruit in the fall so you can avoid winter. And we've been doing some work with him and now on our own to bring that flowering time early enough into the season so that we can flower in July and August and have fruit in August, September, October for blackberries. And the blackberries I brought back there today were picked yesterday. So we're at some of the new selections from the program are in full harvest, and we're getting a very nice product. Yield is still a little bit on the low side, so it's something we're working on, but some yield is better than no yield, which is what we generally get with summer bearing types. So expect to see more blackberry production in the northern climates in the coming years. And they're starting to adapt these types of varieties in California as well for blackberry production. So they can time the plantings to get fruit at a certain number of days after planting on the current year's growth without a dormancy period. And that's really the advance. You don't need dormancy. We'll talk a little bit then. It's very faint. Some of the molecular genetic projects is the other side of the program. So I thought I get asked a lot about, how long does it take to get a variety out? What is the process? So I thought I'd walk you through what we did with Archer, my newest strawberry. And it can be a long process. So the Archer was a selection I made very early in my career here at Cornell, and it was done very much in the traditional sense of things. And so you'll see every step was done through the field on a year-by-year basis, and it takes a long time doing it that way. So we've got some strategies we've worked on, and I'll talk about how we've been speeding that up. But this is how Archer started. So in '99, we made some crosses, made some hybridizations. I didn't have a lot of information to go on. I hadn't worked on berries for very long, but we had the records from the past breeder, and we made some crosses, collected the seed. In 2000, that seed got treated, got planted out into the field. And you just grow them in the field. So we planted them in rows and let them grow. Those have to grow a year, go through winter. The next spring, we get to see fruit. Now we've cleaned that up a little bit, and we use plastic. We put them on plastic to help with weed control, but the process is generally the same. We put them in the ground one spring. The next spring, we get to see fruit. Helps as well. We use a little bit of raised beds when you get a lot of water. This year that didn't happen, but last year we had water. It was so high it went almost over the beds. So from some years we get a lot of rain, and some years you get none, but you can't know that in advance. So in 2001 we made the selection. So this is a 2001 selection. I made some notes that day when I selected it. It's a huge kingberry. Often the primary berry that comes off a strawberry in the industry, they'll call it the kingberry. I put very large Q. Well, I use the term queenberry as well rather than secondary because it's easier to write, but that's not generally used in the industry. And I wrote that it was firm, good interior color, good flavor, tart. That's what I had to go on. I made some notes that day, and I picked 3 fruit. A primary berry, a secondary berry, and a tertiary berry. You add those up, it's almost 100 grams of fruit. So I calculated if you took 100 grams of fruit, which is only 3 berries per plant, on a per acre basis, that's over 4,000 pounds. Each plant produces more than 3 fruit. So this is the kind of thing, when we see this kind of thing, we're like, wow, we got yield right away because we got a lot of fruit size. It comes off very quickly, and it comes off at once. And for us to have a tertiary berry in the 16 gram size is a very good berry in the marketplace. 25 will get you about this big. 50 grams will be about the size of a plum. So it's a very large berry. That's larger than what you normally see. And we'll pick berries down to about 8 to 10 grams is acceptable for a commercial product. So that year we had 1 plant. That's all there is one plant from that 1 seed. So whatever runners it produced, we collected those runners, rooted them, and there's a picture from that first plant. They're not the prettiest thing because I'm out there holding them with one hand, taking a picture with the other. So it's really not, I'm not a photographer. But for me, it gets it in my head so I can look back and say, oh, I remember that one. And that's what we do, to have a picture, kind of a record as we go. You start a plot. It took a year to make the runners, then you plant them again. You got to wait another year before they fruit again. So that takes a year. We planted, and then we look at it for 2 or 3 years because we want to see, is it going to live? Is it going to have some disease that wipes it out in the first, second year? Is the fruit going to grow? Are the plants going to grow? Are they going to produce runners? So we collect more runners from the next plot, and then you plant another plot, and you do it again to make sure, because you got to plant it in different locations to make sure you didn't plant it in the best possible location and it was great, but in a bad location, it's going to die very quickly. So we planted multiple plots in different locations so that we can see this over time. And then you have to evaluate it for several years to see how it comes through winter, to see how the plants grow. Will they persist in the field? Will it have a good yield every year? It takes time to go through this, and it can really take a long time, and during that way, most of the selections get sloughed off. They're found wanting for whatever reason. They're susceptible to some disease. They don't produce enough runners. The fruit quality declines over time. These are things that you see. We take more pictures. So we want to make sure we're planting the same plant every time, and that when it produces fruit, the fruit looks like what we think it's supposed to look like. So each time, each cycle, you're taking pictures to make sure everything is the same. At that point, we decided these are looking good. They're very consistent. We're going to move it into tissue culture, because to take it off the station, we need disease-free plants. I can't collect stuff from the field and send it out to growers or to a nursery, and it could have any kind of pathogen on it. So we move it into tissue culture to clean it up. Make sure it doesn't have any viruses. Make sure it doesn't have Phytophthora, Verticillium, various fungal pathogens. And that can take a few months, but it can take a year or more too. Some varieties don't tissue culture well. So we got plants. We have to check them for virus. We have to produce more plants so we can put them back out in the field. Make sure tissue culture didn't screw it up. Sometimes you put things in tissue culture, what you get out of tissue culture doesn't look like what you put in. And so we have to make sure that we grow plants, make sure they're true to type, make sure they look like what you expect them to look like. All this takes time. And we, of course, take more pictures. We want to make sure it looks like it's supposed to look like. You have to make sure you have the same product that you, in the end, of what you started with. So we confirm trueness of type. At that stage, we're like, okay, we got virus-free plants. Let's send it to the nursery so they can propagate it in large numbers. That way we can get it to some of the growers. So in 2014, we sent out 16,000 plus plants to 11 growers across the state so they could grow it in a large scale and give me some feedback on how it looks for them. How does it look in a commercial setting? Up to that point, we had 25-foot plots at the biggest. So now they get 1,000 plants or 2,000 plants, and you can see it on an eighth of an acre or a quarter of an acre and actually see it in a large scale. They evaluated the first year. They evaluated again the second year, because you want to see, most of the growers will grow it in a perennial setting and they want to see it more than 1 year. So all this takes 15 years. We get to this point and we're finally, okay, it's time to release it, and you're here today. It's being released today. It's being announced. So it is available to growers now. They won't plant it until next year. But of course, we're taking pictures all along to make sure that it's the same thing. Question?
When you send that out to the growers,
yes.
Are they paying for the plants, or do you give it to them?
So what the growers will pay, what the growers will do, or what the nursery will charge is a propagation fee,
Okay.
so that the cost of propagation is offset.
Okay.
So it is not the same price that is, we're not selling the plants, they're offsetting the cost of propagation. It's semantics, but it is not available to commercial growers. In this case, it was only available to growers of the New York State Berry Growers Association through a cooperative agreement with Cornell University that we have with the association. So we didn't send it to just any growers. We sent it to growers who were part of this organization that we had a contract with for doing the testing. And so this was this year. This is fruit I got from a commercial grower. It looked great. He was happy with it. We moved forward so that it's now going to be available. And they will plant the first commercial crop this coming spring in 2017. Of course, we won't see that fruit until 2018. So again, there's time every time you have to grow the plants. So it can really take a long time to get through it. So how do we speed up the process? Well, protected agriculture is one of those ways. Certainly the high and low tunnels speed things up. The plants grow faster. You get a better look at the plants because every year you have a good year because you can control the environment much better. This year in our open field, the raspberries are very, very short. Where they're not irrigated, they just didn't grow much because they didn't get any rain. In the tunnels where it's irrigated and protected, the plants look just like they do every year. They're 8 feet tall and they're covered in fruit. And so we can really get a faster look at things. And since we can look at them quickly, we can evaluate them quickly and move things into the commercial stream much faster because we can make decisions based on better information. We can make that decision quickly and get them into tissue culture quickly. And once we have them in tissue culture, there are commercial labs we work with who can propagate them and propagate thousands of plants very quickly from a few tissue cultures. Both raspberry and strawberry work very well in tissue culture, and so propagation, once you have the culture started, goes very rapidly. And in raspberry, it is commonly done in the commercial nurseries that people will use tissue culture for propagation. The other thing we've done is sped up the grower evaluations. Rather than waiting until that last stage to get it to growers, we're really trying to get it to the growers much sooner. So this year in New York, we sent some 2012 selections to a couple of growers in really preliminary trials, but to get a commercial look at them much earlier in the process, only 4 years from when it was selected versus 10 or 12 years after it was selected. Some of our commercial growers are larger growers, so we do that with the New York State Berry Growers Association. Some of our other cooperators in larger locations, larger industry locations, they want new stuff as fast as they can get it. So sometimes 1 or 2 years after it's selected, they'll get to take a look at it because they're really interested in moving very quickly. And they have the infrastructure to give me feedback very, very rapidly. And so that's one of the things we're doing to try and speed things up. Long term, we'd like to do some marker-assisted selection using genetics and DNA technology to speed up the process and make it more efficient. Throw out the stuff that isn't worthy before it ever makes it to the field. But we're a long ways from that. So one of the things we're doing in the tunnel is growing things in pots. So in the field, when we plant a seedling, it usually has to grow at least 18 months before we can see fruit. The summer types need to grow for 2 years before we can see fruit. These fall types in a high tunnel in pots, we can plant them in the greenhouse in March, bring them out in the pots in May into the tunnels, and they're fruiting now. So between March and now, we get to see fruit, and we can eliminate a lot of stuff that is unsuitable. Now, we can't make great decisions on the stuff that looks best, but we can move it much faster and eliminate a lot of the stuff that we're not interested in. And so this is one of the things that we're starting to do more of. So in a high tunnel like this, it's 24 feet wide, we can fit about 900 potted plants. And so we can go through 900 plants a year in there, and those in the field, those might be in the field for 3 or 4 years for each cycle to look at them. So traditional breeding, as we were doing before, in the molecular genetic side of things, we've done quite a bit of genetic mapping. We've done some marker-assisted selection. We've done some RNA-seq work and genome sequencing to try and increase the level of information that we have for the genetics of raspberry. And for the future, we'd like to try the latest thing that everybody wants to try is CRISPR. But we think there's some really interesting things that could be done in clonally propagated crops with this technology. And since raspberry works so well in tissue culture, it's a fairly natural thing to try it on because it is usually done in tissue culture. So we've done some genetic mapping in raspberry, primarily for Phytophthora root rot resistance. So Phytophthora is a disease of the soil, of the roots that's in the soil. It's pretty devastating on raspberries. It's very persistent. There is good resistance, but most of the resistance are in varieties that aren't particularly useful for the commercial market. So we'd like to do breeding, and you can do breeding, and you can do field analysis, but if it doesn't rain, you don't get Phytophthora. So it's hard to screen stuff in the field for Phytophthora resistance if you have a dry climate, or if you have a dry year like we did this year. We had lots of problems on raspberries this year, but Phytophthora was not one of them because we didn't have a lot of infection. So how do you look at Phytophthora then in a way that is consistent? Well, we developed a hydroponic system so we could actually observe the roots in the raspberry as the disease is developing so that we could get an accurate screen of resistance. And then we developed molecular markers, get a genetic map, and then look for QTL analysis, the quantitative trait. So Phytophthora resistance in raspberry is a multi-gene trait, a quantitative trait, and you look for QTLs to try to figure out where in the genome most of the influence on that trait is coming from. So here's the hydroponic system. You can see when there is no pathogen, raspberry likes it very well. It grows very well. The root systems are white and healthy. But once you introduce the pathogen, it's very quickly apparent which ones are susceptible and which ones are resistant. So it gives us an accurate screen to use for our genetic mapping. And we've been able to use this, and you'll see, multiple times in different aspects of this project. So this work was done by one of my graduate students, Jeremy Pattison, who now works for Driscoll Strawberry Associates in California. And so you can see the genetic map. There's 7 haploid number in raspberry is 7. We have 7 linkage groups. We looked at the disease in different ways. We took a qualitative score, just 1 to 5. Is it diseased or not diseased? And then we looked at stem lesions. So they develop stem lesions as the plant is infected, and you can measure how big those are. Some of the petioles get lesions, so you can do a percentage of the petioles that get lesions, and then you can take a root score as well. Since we could observe the roots directly, when you do a potted study and the roots are surrounded by soil, you get dead or not dead, but it's very hard to tell what's going on in the roots. And so this way we are able to look at the roots directly and get a score there. And we found that all 4 of those traits map to 2 regions. So it tells us that when we're measuring these things, all of them were a good measure of resistance, and that they were measuring essentially the same thing, that same level of disease development. And then the percentage of the variation that was explained by the various scores. So some of them, quite a bit. When you combine the 2 locations, 50% of the variation was defined by that score. Only 25% with a stem lesion, so it wasn't as consistent. You'll see 55 and so on. But these regions of the genome very clearly have something to do with resistance. The problem is that it's when you start looking at the sequence, that's a very large amount of sequence, and so there's quite a few genes in that region that you would have to look at. So we published some of this work in TAG some years ago. It was later confirmed by another group in Scotland that they published a very similar work that came up with the same conclusions, which we were very gratified to see since they're competitors. We have done some marker-assisted selection with those markers in those regions. We screened varieties. We put varieties in there and introduced it and looked at both the markers and the resistance rating from this. And it did pretty well. When we did the marker, it's about 76% accurate. And what we found is that when the resistance came from a different background, the markers kind of broke down. So the background, markers we found followed a resistance from a specific source. And there are other sources in the germplasm that are probably not in the same area, or at least have different markers if they are in that area. When we looked at populations, so seedling populations, it was all over the place. So some of the seedling populations that were of the same source, very, very accurate, 90% or better. But some of the populations that came from sources of resistance that we didn't know, that were not from the resistant Latham, some of them were worse than flipping a coin. So the markers didn't always work across the populations. And that's one of the things we find very commonly in marker-assisted selection, that they're very population-specific, the markers. And so what do? We want to find better markers. That would be a good thing. We continue to do marker work, or we can continue to look at other populations, or we could try to use some other genomic tools. So the marker-assisted selection work, we've presented some of this work at international conferences and published that there. But we wanted to look a little deeper into the question of what is actually happening. What genes are turned on? Why is resistance happening? So we looked at, we wanted to use RNA-seq, or RNA sequencing. We took root tissue from Latham, which is a resistant variety, and Titan, which is a susceptible variety, and we did it pre and post disease. So we wanted to look at what genes were turned on before it had any disease, and then what happened when you introduced the disease into the plant. If you're not familiar with RNA-seq, RNA-seq is a way to capture the RNA at a specific time point. So it isn't really, you're not following it over time, but you grab the RNA from a tissue and isolate it, and that tells you what's going on at that moment. And then in this case, we grabbed it at another moment as well and compared the 2 moments. So you break them up into small pieces, because at the time the technology was, we were at 86 base pairs, or nucleotides was what could be sequenced using Illumina. Turn it into DNA, sequence it, and then you have millions of little fragments, and they have to be all pieced together. Now, there's quite a few computer programs that they do this with. It helps if you have a genome, a reference genome that's already there, because you can reference those sequences straight to the genome. But if you don't, you can put it together just like a jigsaw puzzle where you don't have the picture, and you're just trying to piece it together bit by bit. And of course, it's done with computers. to look at every possible combination to see what lines up. You get more accurate when you have a genome, but sometimes you don't have that, especially with crops like raspberry, where there is no genome published. It's not a widely studied genome or plant, so there really isn't quite the resources you would have for Arabidopsis or rice or corn or one of the more studied crops. We actually did both with that. But so again, we took the hydroponic system. We collected RNA from the roots, which is very difficult to do when they're in a pot covered with soil. So this hydroponic system came back into it so we could collect fresh root tissue and then diseased root tissue. So you could see the difference between the susceptible ones and the resistant ones at the RNA level, at the gene expression level. So we reported on this at PAG and a few other, and the Rubus Ribe Symposium. So what did we do? We had about 71 million reads of 86 nucleotides, which is a fairly typical kind of number. Overall, once we looked at all we identified approximately 17,000 genes that were expressed in these tissues. Of course, all kinds of genes are expressed, and they may or may not be related to resistance. We took between the two groups, between Titan and Latham, and there was a lot of overlap, about 13,000 genes. And then we did an alignment both to a diploid strawberry, which was pretty much the closest genome that we had as a reference genome. And then we did de novo as well, just the one without a picture, when you just try and piece it together based on the sequence you have. This work was done by another graduate student, Judd Ward, also works for Driscoll Strawberry Associate. He is their head of their genome department now. And then we did a differential expression analysis. Look at the genes between the two groups to see what was different. This was the assembly was done how to do it in non-model plants. We published that in the Journal of Botany. Has been very well received because there's a lot of crops that people have very little genome data to work with. They don't have a reference genome when it's like, well, how do we deal with all this RNA-seq data? And so we put that out. So what did we find? We pretty much in both of them, you infect them with Phytophthora and all their defense genes turn on. Everything they have goes nuts. But there's some differential expression. So some of these things were turned on. I won't get into the details of what the genes are, but these have to do with detecting the pathogen and turning on the defense mechanisms. We saw differential expression. So in Titan, they weren't turned on as much, the susceptible one, as they were in Latham. So upregulation of very key defensive genes. The PR genes are pathogenesis related. These are genes that are often turned on and expressed when a plant is trying to fight off a pathogen of some sort. And there's a bunch of these genes. We found 18 different ones that were expressed in Latham. 10 of them were upregulated in Latham, where only 10 were upregulated in Titan, and 3 of them were actually downregulated in Titan. Very different expression pattern of these defense responsive genes between the two types, between the resistant and the susceptible. But of course, these are large numbers of genes that we're looking at. We did a blast search of the data just using the terms salicylic acid, jasmonic acid, systemic acquired resistance, defense, disease. These are all terms that are used to describe genes. And so when we did that, 99 different things came up. Well, 99 is a lot of things to choose from. This is the kind of stuff you get from BLAST. So we could tell, though, these were genes that were actually differentially expressed in the resistant type, so in Latham. So in the control versus the treatment, you can see the number goes up because it's expressed more highly in the resistant, or when the pathogen is actually present. And you see here then these are just the search terms that you use and what pops up from the BLAST searches. And this table went on for page after page after page because of all the information. So you have to sort through all this, figure out what you're working with. So where do you go with this? I mean, 99 is better than 17,000, but we still can't actually do candidate gene work on 99 different genes. It's just not possible with the funding I have and the technology that we have now. It would take a long time and a big team to do it. So some of the things you can rule out fairly quickly. They're very specific responses versus very broad responses. So some of them are about producing lignin in the cells, and that may not have any direct response, but is a kind of a side response to this. So we can start to rule some of that stuff out, and then we want to align it to the genome, to the QTLs that we have, so that we can start looking just in the region where the QTLs are, rather than looking through all the genome, and focus on those first, the genes that might be present in those regions. And that's at the stage where we're at now, analyzing the genome data that we have with the RNA-seq data to try and narrow down our choices. And then once we start figuring out what might be there, that's where the CRISPR would come in. Now CRISPR is something, though, that right now a lot of people don't have a lot of experience with, myself included. But we think there's some targets in raspberry that we can use to learn how to use the technique and make it useful in the future. If you're not familiar with CRISPR, it's a bacterial system that they theorize is used in protecting the bacteria from viral infection, primarily. If you're wondering what the acronym stands for, they're clustered regularly interspaced short palindromic repeats. That's CRISPR. Tells you a lot, doesn't it? They are specific DNA sequences in the bacterial genome that the bacteria can use to target alongside this CRISPR-associated protein 9. It forms a complex, and it can find the foreign DNA in its own genome and systematically or very specifically cut it out and get rid of the pathogenic DNA. That's what we feel. That's what scientists think that the actual system does in nature. What they've done is harness this to take this section here, which is that targeted DNA, and put in one that you want to change yourself. So make this DNA molecule yourself so that you can target a very specific DNA sequence that you want to change. Now, to do that, though, you have to know the very specific DNA sequence that you want to change, and it's a very small piece of DNA that you want to change. So it's really very challenging to get to that point. But my new student, Archana, is working on this project. We're going to do some of this work with thornless, because we think a simple trait that we can very easily visually work with and classify is what we're going to work on. So we have a population that's segregating. We'll do some sequencing to try and find the gene for thornless, and then try to use CRISPR to work on the thornless gene. Because we can do this in tissue culture, we can very rapidly phenotype them as the process goes forward. I think we're at the end of this. In the end, then we want to transfer that to Phytophthora work as we narrow down the different genes that could possibly be interested in Phytophthora resistance. With that, I mentioned a couple of my former students, both working for Driscoll's. Bill Boone was a technician in my program for many years, and Colin is my current technician. Without him, I don't have a chance to get a whole lot done. And of course, Archana will be working with me for the next several years on this CRISPR project. We're very excited to see where that goes. So, in a nutshell, there's the berry breeding at Cornell. And if there are any questions, I will be around for a few minutes. So a question about me not mentioning organic. Organic is a very challenging thing for berries because they're very, very perishable. They have a lot of things that like to eat them besides humans. The York's even more difficult because of all the rain. So without the use of fungicides, it's very challenging to get organic. This new pest called spotted wing Drosophila, which is a little fruit fly, is causing even more trouble with it. Most organic production right now is grown in dry climate locations, and they're using organic pesticides to try to control that fly. But that's the only approach we have right now. Nutrition is very difficult as well in these tunnel systems with organic fertilizers because they don't work well in the fertigation systems. So we don't do a lot of organic raspberry or strawberry production in New York just because of the challenges of the environment. Question?
I'm just curious if you looked at the nutritional profiles or any phytochemical profiles of the different raspberry colors.
The question was about looking at phytochemical and nutritional differences amongst the varieties. We have done a fair bit of work in that area, both here and then in collaboration with North Carolina State, where they have a lab that does a lot of that work. We find wide ranges of certainly anthocyanin content, phenolic content amongst the berries. They're all very high though. Even the yellow ones are very high in phenolic compounds, even though they have no pigment. They're high in vitamin A, folic acid, vitamin C, a host of things. In general, berries are good for you. We want people to eat more of them, not necessarily specific ones. So, but there's a lot of published work on the differences amongst the varieties. Any questions in Geneva? I've kept you there long enough.
Courtney, this is Bruce here. In your RNA-seq work, how did you choose a time point to analyze and a time point that is a time point after inoculation to collect your samples? And would there be any better plant material if you were to do this again?
So, in our studies, I think you all heard the question. In our studies before, in doing the analysis of inheritance, we were able to observe the root systems as the pathogen was introduced and the disease progressed. And so we used our experience from that to choose a time point. Well, actually, we did 0 time point before the pathogen, and then we actually took samples at 2 days, 6 days, and 30 days. And those were very distinct time points in the inheritance study. At 2 days, there was no symptoms yet, but there was infection. And so we could measure that there was infection in the plant. At 6 days, you start seeing symptoms. And at 30 days, you start seeing growback of the roots that are in the resistant plants. So we didn't have that last time point susceptible ones because they were all dead. But we were able to then choose those points so that we could see the progression of the disease, not only in the plant, but in the expression. As far as a better tissue, it seems that for Phytophthora that the resistance is in the root system, is expressed in the root system, so I think that's the best tissue to use, though it is somewhat challenging to use.
Courtney, I'm asking about the varieties that you would use. Would you use the same varieties? Are these varieties related to each other, or are there any others that you would consider using?
At this point, we want to stay with the varieties that we've been using because we have so much historical data on those varieties. Certainly, there are other sources of resistance that we would like to look at in the future, but you can only do so much because we know there are sources that are not related to this source that we're using. But at this point, we don't have plans to use other genotypes.
So our time is up. I want to thank Professor Weber for a fascinating presentation.
This has been a production of Cornell University, on the web at cornell.edu.